have already studied. You have already studied the microsporogenesis, megasporogenesis, microgametogenesis, and megagametogenesis. You have already studied the development of female gametophyte or embryosac in the previous lecture. In the previous lecture, today we have to study the different types of uh, embryosacs. The types of embryosac. The embryosac, which is also known as the female gametophyte, develops from the functional megaspore. You all know that out of a megaspore tetrad, only one megaspore cell remains functional, whereas the other three megaspore cells undergo degeneration. And it is also known to you that it is either the chalazel or the micropylar megaspore which remains functional. Okay? So this is, and this functional megaspore represent the first cell of female gametophytic generation, and it gives rise to the development of the female gametophyte or the embryo sac. And this embryo sac is a very important structure, and this embryo sac is of uh, embryo sacs are of main embryo sacs are of three main types, which include: number one, monosporic embryo sac; number two, bisporic embryo sac; and number three, tetrasporic embryo sac. These three types of embryo sac are these three types of embryo sacs depend upon the number of the megaspore nucleus or nuclei taking part in the development of embryo sac. If a single megaspore nucleus is taking part in the development of embryo sac, the embryo sac is known as the monosporic. If the two nuclei are taking part in the development of the embryo sac, the embryo sac is known as bisporic. And if all the four nuclei take part in the development of embryo sac, the embryo sac is known as the tetrasporic type. You know that the megaspore mother cell undergoes reduction division to form four nuclei, and there is four nuclei, and if there is reduction division without the development of any cell plate, all the four nuclei will remain in the same cell, remain there, and if all these four nuclei participate in the development of the embryo sac is known as the tetrasporic embryo sac. If the two nuclei take part in the development of embryo sac, the embryo sac is known as the bisporic. And if a single mega, as you know that out of the four megaspore, it is only one megaspore which remains functional, and the three megaspore cells undergo degeneration. So in this case, the nucleus of the functional megaspore, a single nucleus of the functional megaspore, gives rise to the development of embryo sac, and such embryo sac is known as the monosporic embryo sac. All these three types of the embryo sacs have their subtypes. What does it mean? It means that monosporic embryo sac is of two types, which include: number one, 8 nucleate polygonum type of embryo sac, and number two, 4 nucleate Oenothera type of the embryo sac. Similarly, the bisporic and tetrasporic embryo sacs are also of subtypes, which we will be studying in the next lecture. But today I have to study the monosporic type of embryo sac in detail, and I have just made you know that the polygonum and Oenothera type of embryo sacs are the two types of the monosporic embryo sac, and I have also described the process and the number of divisions which take place in the megaspore to form the polygonum or Oenothera type of embryo sac. Now let us study these two types of the monosporic embryo sac in detail, and I am just going to show you the whiteboard where the diagrams of the 8 nucleate polygonum type and diagram of the 4 nucleate Oenothera types are sketched there for for making you understand. I just have drawn this structure to make you know. So now look at the whiteboard and try to understand. Try to understand the first type, polygonum type, and the second one, that is the O Enothera type. Look at the whiteboard. Now I am going to show you the whiteboard. This is the whiteboard, and on which the diagrams has been sketched. Look here. The embryo sacs, I have written here the embryo sac and types. We are studying today the types of the embryo sac. The embryo sacs are of the three major types: monosporic embryo sac, bisporic embryo sac, and tetrasporic embryo sac. Today, in case of monosporic embryo sac, a single megaspore nucleus participate in the development of the embryo sac. In case of bisporic embryo sac, two nuclei participate in the development of embryo sac. In the tetrasporic embryo sac, all the four haploid nuclei participate in the development of the embryo sac. Now let us see the monosporic embryo sac and its type. This monosporic embryo sac is of two types. Number one, 8 nucleate polygonum type and 4 nucleate O Enothera type. Now let us study the first one, which is 8 nucleate polygonum type of embryo sac. This polygonum type of embryo sac develops from the chalazial megaspore, not from the micropylon. This 8 nucleate polygonum type of embryo sac develops from the chalazial megaspore. The megaspore nucleus divides thrice. The megaspore nucleus divides thrice to form 8 nuclei. 4 plus 4. 4 nuclei in the chalazial side and 4 nuclei in the micropylon side. Presence of 3 antipodal cells. Because the 3 antipodal cell, 2 polar nuclei and 1 egg apparatus. Look here. This is the diagram to show you how the cell is divided, how the this 8 nucleate embryo sac is being formed. Look here. This is the megaspore. This is a functional megaspore. This is a functional megaspore, and it has a haploid nucleus. This nucleus of this functional megaspore divides first to form 2 nuclei, one toward the chalazial side and one toward the micropylon side. Again, these 2 nuclei undergo the second division. That means divides next time to form 2 nuclei in the chalazial side and 2 nuclei in the micropylon side. Again, these 4 nuclei undergo third division to form 4 nuclei at the chalazial side and 4 nuclei at the micropylon side. Now 8 nuclei have been formed. And now there is how these nuclei are arranged to form the embryo sac. Look, the three out of these 4 nuclei, one nucleus moves in the center and 3 nuclei remain at the top, at the chalazial side. Similarly, 3 nuclei from the lower side, micropylon side, remain there, and 1 nucleus comes in the center. So in this way, look here, the 3 nuclei at the chalazial side form 3 antipodal cells. The 2 nuclei coming one from the chalazial side and one coming from the micropylon side form the polar nuclei, and these polar nuclei are not attached together. Rather, they remain distinct from one another. They remain in pair. Therefore, we can designate their formula N plus M. And the lower side at the micropylon side, there is presence of an egg apparatus in the which consists of a centrally located egg and 2 laterally located synergids. So this is the, if you are asked in your examination to describe different types of embryo sac, you will have to write in this way. You will have also to sketch the diagrams, such diagrams there. Without diagram, you are not going to get the marks in the examination. The diagrams are very important if you are answering this question types of embryo sac. So this is the polygonum type, which is called the typical embryo sac, and this is one of the two types of monosporic embryo sac. We have already seen this 8 nucleate. Now look at the 4 nucleate O Enothera type. This is the 4 nucleate O Enothera type of embryo sac. Look here. This is the, in this case, the micropylon megaspore is functional. This nucleus undergoes first division to form two nuclei. Again, these two nuclei divide to form four nuclei. In this case, the third division, this nuclei and these four nuclei do not undergo any further division. In this way, only four nuclei are produced. Therefore, it is called four nucleate. And out of these four nuclei, three nuclei at the micropylary end form the egg apparatus consisting of an egg and two synergids, and remaining one nucleus remain in the center to form a single polar nucleus. So what are the features of this Oenothera type? Oenothera type of embryosac develops from the functional micropylar megaspore. Third division does not take place. Absence of antipodal cell. There is complete absence of antipodal cells. Presence of a single polar nucleus. Presence of an egg apparatus. One egg, two synergid. Found in the in which family this such type of embryosac is found. This is found in the members of family Onagraceae, and it was for the first time reported by Hofmeister in the members of Onagraceae. Today we have already studied the monosporic type of embryosac. And these two types are 8-nucleate polygonum type and 4-nucleate Oenothera type. And this 8-nucleate polygonum type is a typical embryosac. Am I clear to all of you? Am I clear to all of you?
Yes, sir.
Yes, sir.
बात समझ में आई कुछ?